This is the third lecture in a series of lectures on the introduction of immunology. This lecture focuses on T lymphocyte activation and the development of T lymphocyte effector function. The learning objectives for this lecture are to describe the structure and function of what are termed the primary and secondary lymphoid organs of the immune system, and briefly describe how lymphocytes and antigen circulate through secondary lymphoid organs and how antigen and lymphocytes interact with one another. And 3, to describe the properties of an adaptive immune response, in particular the specificity and memory characteristics of an adaptive immune response, and to describe the difference between naive and effector memory lymphocyte subsets, and finally to describe the major effector functions of T lymphocytes. So I showed this slide in the previous mini lecture, but again to emphasize the importance of antigen receptors in these two phases of lymphocyte development and function that are defined by antigen receptor expression. So this first phase is illustrating how lymphocytes develop from a common lymphoid progenitor into a large number of different individual lymphocytes with different individual antigen receptor specificities. And these lymphocytes go out of these primary lymphoid organs and reside in secondary lymphoid organs where they're activated upon encounter with antigen. And then depending on the antigen receptor specificity of the individual cell, these cells will proliferate, make more of themselves, and differentiate to become an immune effector cell. And some of these cells will become very long-lived and become a long-lived memory cell, able to respond secondarily to antigen encounter in the future. And so just to review the difference between primary and secondary lymphoid organs, primary lymphoid organs are defined as lymphoid organs where lymphocytes develop. And so there's two main primary lymphoid organs. There's the bone marrow, where B cells develop, and then there's the thymus, which is in the mediastinal cavity, where T cells develop. So T for thymus, B for bone marrow. And this is just a picture showing you that there's a relatively rare congenital disease called DeGeorge syndrome, where one of the congenital defects is associated with failure to develop thymic epithelial cells and thus failure to develop a thymus, which leads to a lack of T cell development. The thing to remember really about all immune system functioning is that immune cells closely interact with one another. And this is just a scanning electron micrograph showing you thymocytes, so developing T cells in the thymus, and how closely they're associated with each other and other cells in the thymus, as well as extracellular matrix interactions. And these cellular interactions and extracellular matrix interactions provide signals to the developing cell and are really critical in determining their cell fate, whether or not they survive and what kind of T lymphocyte they develop. For example, whether or not they develop into a CD4 T cell or a CD8 T cell. So similar interactions occur really in all lymphoid organs. Cells are in close proximity to one another, and interactions are facilitated by stromal cells like fibroblasts, connective tissue, and soluble molecules like chemokines. B cells develop in the bone marrow, again, B for bone marrow, and conceptually development is very similar to what's happening during T cell development in the thymus. The main job during both B and T cell development is to develop into individual cells that express a mature form of the antigen receptor. And that's what's being shown in this slide. So for both B cells and T cells, in this case for B cells, you have a lymphoid progenitor cell that does not express an antigen receptor, but it undergoes somatic recombination or antigen receptor gene rearrangement, as I mentioned in the previous lecture, on the way or on the path to becoming a mature B cell, where these cells will express the mature form of the B cell antigen receptor. And after this process is complete, and only after that, will these cells then leave the primary lymphoid organs and seed secondary lymphoid organs like lymph nodes or spleen or mucosal-associated lymphoid tissue. So, after development in primary lymphoid organs, lymphocytes migrate to secondary lymphoid tissue. Secondary lymphoid tissue is largely defined as spleen, lymph nodes, and tissue that's associated with mucosal sites or barrier sites, especially in the gut and the respiratory tract, and to a certain extent in the reproductive tract. So first, to focus on lymph nodes. Lymph nodes are present throughout the body in different regions, and circulation is complex of antigens and lymphocytes in the lymph nodes. So there's two things that happen. So one is antigen gets drained from barrier sites through efferent lymphatic vessels that go into the lymph node. And essentially the lymph node is a place that's filtering antigen that's received from barrier sites. At the same time, lymphocytes are recirculating throughout the body in different lymphoid organs, and the lymphocytes get into the lymphoid organs through the bloodstream. So antigens get in through these efferent lymphatic vessels, lymphocytes get in through the bloodstream, and then they may interact or they may not, as you'll see in a few minutes. And then the lymphocytes will then exit the lymph node, in this case, through the efferent lymphatics and recirculate to other lymphoid tissue in the body. The spleen is really conceptually similar. Again, it's a secondary lymphoid organ, but instead of filtering lymphatic fluid, it's filtering blood. But similarly to lymph nodes, it's a place where lymphocytes meet antigen and interact with each other, and that's where an immune response is initiated in these secondary lymphoid organs, like the spleen, like the lymph node. The final type of major lymphoid tissue is lymphoid tissue that's associated with mucosal sites. The biggest chunk of this kind of lymphoid tissue is associated with the GI tract. And you can see the GI tract takes up a huge amount of surface area in the human body, much bigger than the surface area of the skin, because you have all this coiled up intestine that has a large amount of interaction with antigens that come in through the GI tract. And just to kind of digress a little bit about what's going on at this particular barrier surface, so there's lots of exposure of different antigens, and this can include commensal bacteria, as I mentioned in the very first lecture, that normally inhabit these areas, as well as pathogens that may come in, as well as things that you ingest, like food. And all of these types of antigens will interact with mucosal epithelial cells. And just within these epithelial cells, or just beyond the epithelial barrier, there's a whole variety of different immune cells, including T lymphocytes and B lymphocytes, but as well as innate immune cells like macrophages, neutrophils, eosinophils, mast cells, dendritic cells. And all these cells interact with each other and interact with antigen that are coming down into this barrier site to help mediate both protection against harmful pathogens, but also tolerance of non-harmful pathogens so that we don't develop inappropriate inflammatory reactions. Just to emphasize again, the GI tract is an incredibly rich area of immune cell activity and immune activity, and some estimates are that approximately 40 to 60, or some estimates 80% of all your total lymphocytes in your body reside in one way or another at these mucosal sites. And this is just a schematic showing you the different types of lymphoid cells and organs that are associated with mucosal tissue. So this is the GI tract, and so you have intestinal villi here, and epithelial cells, and you have immune cells that are actually exist in between epithelial cells here, and also immune cells in the lamina propria, just underneath the epithelial barrier. And you also have organized areas of lymphoid tissue just underneath the epithelial cells that can exist as isolated lymphoid follicles, so a T cell and B cell areas, as well as Peyer's patches that are kind of an agglomeration of multiple T cell and B cell areas. And so all of these secondary lymphoid organs serve as places where lymphocytes can interact with antigen. All the time we have lymphoid cells recirculating throughout the body and throughout these different lymphoid tissues. And this is just a cartoon showing you one lymph node in the popliteal space behind the knee here, and how you have lymphocytes coming into this lymph node through the bloodstream to scan the lymph node microenvironment for antigens that will engage its antigen receptor. And if it doesn't see anything, it will leave the lymph node, exit through the efferent lymphatics, and rejoin the circulation and kind of enter into different lymphatic tissue in other anatomical sites. However, in this case, if you stepped on a splinter, you might have drainage of antigens associated with that splinter through the afferent lymphatics into the lymph node. And if you happen to have a lymphocyte that's in the lymph node at that time that has a receptor specific for that antigen, that lymphocyte will become activated and help mount an immune response against the antigens associated with that splinter. And so let's talk a little bit about the steps that are involved with generating that initial adaptive immune response. And so, as I mentioned before, it really relies on the interaction between cells and factors of both the innate and adaptive immune systems. And so this is a slide that I showed in the very first mini lecture, again showing that what happens when you get a splinter. You break barrier surfaces, you break the skin, you may introduce bacteria into that site. Those bacteria will engage with resident innate immune cells like macrophages. These macrophages will become activated because of activation through their pattern recognition receptors that will result in the production of molecules like cytokines and chemokines that act on the surrounding tissue to help promote the influx of fluid and cells and other molecules into the site of the inflammatory response. But the next step in this process is that this inflammatory response will then lead to the activation of an adaptive immune response, meaning activation of T lymphocytes and B lymphocytes. And one of the first key steps involved with this involves the activation of another innate immune cell that's constitutively present at these barrier sites called the dendritic cell. And so the dendritic cell will sample the microenvironment that it's in, and if its pattern recognition receptors are activated, it will signal these cells to migrate from the barrier site down through afferent lymphatic vessels, and it will take with it antigens that it's taken up from that site. So it will bring antigens that it was exposed to at this site down into the draining lymph node. And so here it's coming down through the afferent lymphatic vessel into the parenchyma or tissue of the lymph node. And if you have a lymphocyte, in this case a T lymphocyte, that has a receptor that's specific for antigens that are being presented by this dendritic cell, and remember these antigens are going to be presented by MHC molecules. So this dendritic cell is a professional antigen-presenting cell that's processed and presented peptides from antigens that it's encountered at the barrier site, and is presenting those peptides in a form that this T cell can recognize. And so if you have a T cell that has a receptor specific for those antigens, these T cells will clonally expand. They'll proliferate and make more of themselves. And one of the things that will happen, and we'll talk more about in a few minutes, is that these cells not only will clonally expand, but they will differentiate into developing effector function, and they'll then leave the lymph node, feed back into the peripheral tissue, potentially where the inflammation is happening, and further activate an immune response against those antigens at that inflammatory site. And so this is a kind of a cartoon of this key interaction between this professional antigen-presenting cell, a dendritic cell, with the T cell. And so remember, as I said, that in order for this T cell to become activated, its T cell receptor has to engage with MHC peptide complexes presented by the antigen-presenting cell. And the second important part of this process is that this antigen-presenting cell also has to express what are referred to as co-stimulatory molecules that will engage with partner co-stimulatory molecules expressed on the surface of the T cell. And the most important co-stimulatory molecule for initial T cell activation is a molecule called CD28, which engages with another molecule on the surface of the antigen-presenting cell called B7. And the combination of signals that the T cell receives through the CD28 molecule and signals received through the T cell antigen receptor will result in T cell activation and will result in proliferation and expansion of that cell and differentiation into an effector cell. And the other important thing to remember from this slide that's really critical for activating an adaptive immune response is in order for this antigen-presenting cell to be very good at presenting antigen to T cells, it had to be activated first by its pattern recognition receptors as a consequence of it engaging with pathogen-associated molecular patterns or damage-associated molecular patterns that interact with pattern recognition receptors like toll-like receptors that I talked about in an earlier lecture that allow this cell to produce cytokines and to express high levels of these costimulatory molecules like B7 and facilitate this cell's ability to then interact and activate this T lymphocyte. And so this is really a central concept in adaptive immune cell activation and feeds back always to the specificity of the antigen receptor that the lymphocyte makes. And this model of an adaptive immune response is referred to as the clonal selection theory. And what that refers to is that because each individual lymphocyte tends to express an antigen receptor of a distinct antigen specificity, the antigen receptor specificity defines that lymphocyte as a clone. And remember, antigen receptors are expressed prior to antigen interaction. The repertoire of T cells and B cells with different antigen receptors occurs randomly during T cell and B cell development in primary lymphoid organs. So after these lymphocytes go out into secondary lymphoid organs and they encounter antigen, the antigen selects the lymphocyte based on the antigen receptor specificity of that lymphocyte. And then in this case, it would be the yellow lymphocyte that has the yellow antigen receptor, engages with antigen, and it signals the cell to proliferate, to make more of itself, and to differentiate into an effector cell. And it's then these effector cells that eliminate antigen. And so this clonal selection theory is really a nice way of explaining a lot of things that characterize the adaptive immune response. So one thing it explains is the specificity of the adaptive immune response, because it indicates that only cells that have a receptor for this specific antigen are going to get activated. And cells that don't have that receptor specificity, cells that have other antigen receptor specificities, do not become activated. And so you have a very specific and focused immune response. And the specificity of the immune response is really illustrated on curves like this in a nice way. And so what's shown here is on the x-axis timing of the immune response, and on the y-axis a measure of a biomarker of an immune response. And what you can see here is when somebody is exposed to a given antigen, in this case antigen A, the first thing that happens is you get an initial immune response. And that initial immune response is referred to as the primary immune response. And that refers to initial activation of immune cells, lymphocytes, that have receptors for antigen A. And what you can see on this curve is that the immune response, this primary immune response to antigen A, is relatively slow. It takes a long time to develop and is relatively small and decays within a week or two. Okay? And the reason it's slow and small is that the precursor frequency of lymphocytes bearing a receptor for any individual antigen is relatively low. So for example, on the order of 1 in 100 cells may have an antigen receptor specificity, let's say specific for an antigen associated with the influenza virus. And what this means is that the first exposure to an antigen is often not sufficient to generate a decent immune response that's associated with the primary immune response to control the antigen, because it takes a long time to develop and because it's relatively weak. But what you also notice on this curve is that upon repeated exposure to the antigen, in this case again antigen A, the second immune response to antigen A is much quicker. It happens more quickly than the initial primary response to antigen A, and it is also much larger in magnitude. And the reason that it's quicker and larger in magnitude is in part because the precursor frequency of cells that are specific for the antigen is increased, and it's increased because of the initial expansion during the primary immune response. And so because the precursor frequency is increased, the response the second time you see an antigen is more rapid and more effective. And so this really summarizes two of the key features of an adaptive immune response. It illustrates that the adaptive immune response is very specific for a given antigen, and also that it has what's referred to as immunological memory, meaning that the second time that you see that same antigen, the response is faster and larger in magnitude, and usually more effective in clearing that given antigen. And the final feature that is characteristic of an adaptive immune response is that the second time that you see antigen, that cells will exhibit a different effector function or a more developed effector function than occurred during the initial primary immune response. And so let's talk a little bit more about the different effector functions associated with T lymphocyte activation. So this is a cartoon just illustrating the fact that a naive T cell, a T cell that's never seen antigen before, or a virgin T cell, can develop into different types of effector functions depending on the type of T cell it is and the types of signals it receives as it's being stimulated. And so here you can see on the left is that CD8 T cells, so T cells that happen to express the CD8 protein. When they're stimulated, they are often predisposed to develop what's referred to as a cytotoxic T cell effector function that can kill cells that are infected with viruses or other types of intracellular organisms. And so if the CD8 T cell develops the cytotoxic effector function, it can then kill in this case, the virally infected cell and help clear the virus. CD4 T cells can develop into a bigger array of different types of effector phenotypes or effector functions that are classified as Th1, Th2, Th17, and in this slide, T regulatory cells. And these different types of T helper effector functions are associated with different types of inflammatory responses or different types of immune responses, as I'll talk about in a few minutes. But this is nicely summarized on this slide for a few of these effector responses. So for example, T helper 1 effector cells are especially good at engaging with macrophages and activating macrophages that are phagocytose intracellular bacteria. So the host immune response that is most efficient at dealing with bacterial infections that are phagocytosed by macrophages is typically a T helper 1 type effector response, because these types of T cells make cytokines that will help these macrophages have better bacterial static and bactericidal effector function to kill the bacteria that is phagocytosed. Another T cell effector function is a Th2 effector function, and T helper 2 effector cells are especially good at making cytokines and interacting with B lymphocytes, and it will facilitate B cells to proliferate and to produce antibodies that in turn can help protect against different types of infections, especially extracellular infections where the antibodies have access to those microorganisms. Another more recently described T helper effector cell subset is a helper cell subset that's referred to as T helper 17 cells. And what these cells do is again make cytokines like interleukin 17 that activate other cells like fibroblasts and epithelial cells that in turn activate and recruit neutrophils into the site of T effector cell activation and help those neutrophils phagocytose and clear certain types of extracellular bacteria and fungi. And then finally, another type of T effector cell has actually more of a regulatory or suppressive effect. And it's very important when you're thinking about immune responses is not just to think about how to turn on an immune response, but also how to turn off an immune response or to prevent inappropriate immune responses from happening in the first place. And what T regulatory cells are a specialized type of T effector cell that instead of activating immune responses, it actually downregulates or inhibits immune responses and helps protect against inappropriate activation of the immune system and helps protect against certain types of autoimmune disease. And so whether or not a given naive T cell develops into these different types of T effector cells depends to a large extent on the types of signals that the T cell receives in the microenvironment, in the lymph node, or in peripheral inflammatory sites. And so you can think of these signals that the T cell receives at these sites as kind of three main types of signals. So one signal is received by the T cell antigen receptor itself when it interacts with antigen-presenting cells that are presenting MHC, in this case MHC class II peptide complexes. Another type of signal are the signals received through co-stimulatory molecules. As I mentioned, an important co-stimulatory molecule for initial naive T cell activation is the CD28 molecule, engaging with B7 molecules expressed by the antigen-presenting cell. And the third type of signal that the developing T cell may receive during an immune response are cytokines in the microenvironment that also influence what type of effector cell subset the naive T cell will develop into. And so this combination of extracellular signals influence all aspects of T cell activation, including proliferation, whether or not these cells will survive, how many of them may develop into long-lived memory cells, and also the types of effector cells that they'll develop into. And so this development of effector function is something that's relatively slow. It takes days, and it can be regulated at multiple stages. So it can be regulated at this initial stage where this initial naive T cell is being activated by T cell receptor engagement with MHC peptide complexes. And in this case, this is a T cell that's a CD8 T cell, so it's a T cell receptor recognizing MHC class I peptide complexes, and it gets a signal through its T cell receptor. It's also getting a signal through its co-stimulatory molecule CD28. And the combination of those signals allows this cell to proliferate, in part because it promotes the production of a cytokine by this cell called interleukin-2 that allows this cell to proliferate and make more of itself. And not only that, but allows it to differentiate. And in this case, this is a CD8 T cell, and so it's likely to differentiate into a cytotoxic T cell over time, something that happens days later. And so after activation and differentiation and proliferation, some of these cells will migrate into extracellular sites and seek out cells that are infected with the virus that contributed proteins that initially activated this cell. So in this case, imagine this is a T cell that's specific for hepatitis B, and it's migrated to the liver where it finds hepatitis B virally infected hepatocytes, and it will engage with MHC molecules expressed on the surface of those MHC expressing hepatocytes, and that will signal the cytotoxic T cell to release cytotoxic granules that will specifically kill the cell that it's physically interacting with. So it'll kill this cell, it'll kill the hepatitis infected cell, but it will not kill neighboring cells that are not infected with the hepatitis virus. And so again, this is a very specific immune response, but it's slow in developing. It takes several days for the naive CD8 T cell to develop that cytotoxic effector function. And so an important concept here is I just want to emphasize yet again, is that these naive cells or virgin cells, when they initially interact with an antigen-presenting cell, they don't have that effector function yet. They haven't developed, they haven't differentiated into these effector cells. They need time to differentiate, to change their pattern of gene expression that allows them to develop effector activity upon the next time they're stimulated. So in the case of a cytotoxic T cell, it takes time for them to express the proteins that allow them to kill the virally infected cells upon secondary stimulation. So let's talk just a little bit more about CD4 effector cell subsets. So I already mentioned these four major classes of T effector cells: Th1 cells, Th2 cells, Th17 cells, and T regulatory cells. And so one way that these cells are defined is by the types of cytokines that they make that confer different types of immune effector functions. So for example, I said that Th1 cells are especially good at activating macrophages. And that's because Th1 cells make lots of interferon gamma that engages with receptors expressed by macrophages that facilitate the immune function of macrophages and allows those macrophages to be better able at killing organisms that it's phagocytosed. Whereas Th17 cells are characterized by their ability to make this other cytokine called interleukin-17 that ultimately results in activation and recruitment of neutrophils, whereas Th2 cells make yet other cytokines, predominantly interleukin-4, interleukin-5, interleukin-13, and these cytokines are especially good at activating B cells to make B cells make certain types of antibodies that we'll talk about when we talk more about B cell antibody activity, but also make other cytokines that are good at activating characteristic inflammatory responses that are associated with allergic inflammatory responses, like eosinophils and basophils and mast cells. So Th2 effector responses are often associated with allergic type inflammatory responses, whereas Th1 effector responses are associated with different types of inflammatory responses that are dependent on macrophage activation. So one way that regulates the development of the different T effector cell subsets is it correlates with the type of pathogen that is activating the immune response in the first place. And so, for example, some viruses and some bacteria will engage with pattern recognition receptors on cells in the microenvironment in that infection, for example, on dendritic cells or macrophages. And those pathogen-associated molecular patterns will promote the activation of these cells to produce characteristic cytokines like interleukin-12, which in turn promote the development of a Th1 type effector response that evolutionarily is developed so that it's most efficient at dealing with those types of viral infections or bacterial infections. And so the type of immune response often correlates with an effective immune response against a given pathogen. And so there's this kind of interaction between the pathogen, the innate immune system, and the adaptive immune system that dictates what type of immune response or inflammatory response is generated. Another example of this is during infection with large multicellular worms. These worms produce products or pathogen-associated molecular patterns that help promote the production of other cytokines like interleukin-4, which in turn promotes the development of a Th2 type effector response that's associated with a different inflammatory pattern that's thought to be more effective at dealing with those types of infections. So what I want to do now in this movie is just kind of review a lot of things that we've talked about over the previous couple of lectures and earlier in this lecture today that incorporate both activation of the innate immune response and activation of the adaptive immune response into a big picture.
An immune response involves events that unfold both locally at the site of an infection and at more distant sites, such as nearby lymph nodes. We can see the integration of the different parts of the immune response if we follow the course of a typical infection. Most pathogens are kept outside of the body by epithelial barriers, such as the epidermis, and are crossed only when there is an injury or tissue damage. After an injury, bacteria cross the epidermis and establish an infection in the underlying tissue. Phagocytic cells in the tissues, such as macrophages and neutrophils, engulf the pathogen. Dendritic cells are also phagocytic and are activated by binding pathogens to leave the site of infection and migrate to a lymph node. The migrating dendritic cells enter the lymphatic vessels and are collected in a draining lymph node. In the lymph node, T cells are activated by antigen presented by the dendritic cells, and in turn activate B cells to secrete antibody. Effector T cells and antibody molecules return to the circulation. They leave the circulation again at the site of infection, where inflammatory mediators have induced changes in the blood vessel endothelium. CD4 T cells activate macrophages to become more cytotoxic, while antibody recruits complement to lyse bacteria directly and to opsonize them, enhancing their uptake by phagocytes. In the case of a viral infection, activated CD8 T cells would kill any infected cells present.
So that nicely summarizes a lot of the events that we've gone over that are important for initiating the initial development of an immune response. And this is just a curve showing you the same type of thing in terms of a T cell engaging with an antigen-presenting cell, and these cells will clonally expand or proliferate and develop effector function. But what we haven't talked about yet is how this immune response is regulated over time. And one of the things that you can see in this curve is that you have an initial development of this immune response and development of effector function, but over time, this immune response decays or goes away. And it goes away because there's a variety of active regulatory mechanisms that turn off the immune response. These are turned off by molecules that are expressed by activated cells that are often referred to as negative costimulatory molecules, and examples of that would be a molecule called CTLA4, another molecule called PD-1. And then the other thing that happens is a lot of these immune cells that get activated, these immune effector cells, also die by apoptosis. Because either of removal of the antigen or because of active engagement with death receptors with other cells that result in apoptosis of those cells. But then at the end of this process, there's usually a few surviving or relatively fewer number of surviving cells, and these are the cells that are expanded in number compared to the initial primary T cell receptor repertoire and would take part in a secondary or memory cell response. And so these are the cells that have differentiated and expanded and are better able to mount a quick, rapid, effective immune response upon secondary stimulation. And so just to summarize these different events in the life of a T cell, just very quickly, naive T cells require survival signals from, in this case, self-MHC and self-peptide molecules, but ultimately they will encounter a non-self antigen that will result in overt activation and proliferation of these cells. These cells become activated, and some of them will develop into effector cells and mediate an immune effector response. Some of those cells will become long-lived memory cells, and so they're better able to respond secondarily when they are subsequently exposed to antigen.